Now we'll move on to the second topic and the second speaker, which is Matteo Sensi from Università degli Studi di Modena e Reggio Emilia in Italy. And Matteo will speak about the development of an EGOFET immunosensor for the detection of anti-drug antibodies. So you can turn on your mic and video just in case, and I will put the lecture on.
Hi. Hello to everyone.
University of Modena and Reggio Emilia. Today, I'm going to talk about our project on the development of an EGOFET immunosensor for the detection of anti-drug antibodies. Our vision is to help the immunotherapy by monitoring the immunoresponse against the drug itself. Our mission is to develop new, fast, and cost-effective point-of-care devices to detect anti-drug antibodies which are generated in patients treated with immunotherapy. As a model system, we choose to study the ADAs against nivolumab. Nivolumab is a checkpoint inhibitor which binds the surface protein PD-1. PD-1 downregulates the immune system when it's bound to the ligand PD-L1 and 2 expressed on the surface of cancer cells. In this way, cancer cells can evade the immune system, but when nivolumab is bound to PD-1, it hinders the interaction between PD-1 and PD-L1 or 2, and so the cancer cell is attacked by the immune system. But the immune system also develops anti-drug antibodies against the drug nivolumab, so reducing the efficacy of the therapy. It's crucial to develop devices able to detect the presence of anti-drug antibodies in patient samples, both to avoid the reduction of the therapy efficacy and also possible negative side effects on the health of the patients. Actually, the solutions to detect others against nivolumab are mainly ELISA kits, who can allow a qualitative detection, or chromatographic techniques. All of these techniques require labels and a specialized setup. In our approach, we used electrolyte-gated organic field effect transistors, EGOFET, which are label-free devices in which the sensing and amplification step happens at the same time. This is possible thanks to the intrinsic properties of organic semiconductors. In these devices, we have 3 electrodes, a gate electrode, where is functionalized the probe, in our case the drug itself, nivolumab, thanks to the binding with the protein G, and the other 2 electrodes called source and drain are interdistributed and covered by a semiconductor field. Between the semiconductor and the gate electrode, there is a drop of sample. By applying a voltage between gate and source and drain, we can obtain a current between source and drain, which is modulated by the voltage of the gate. By modifying the gate, so by binding molecules, proteins, or other analytes, the modulation of the current between source and drain is changed, and so this small difference is amplified by the property of the semiconductor itself. In this way, it is possible to detect low analyte concentrations with a low power consumption. Why we choose organic electronics? Because it's a label-free method, these devices are sensitive, are cost-effective, since we can print by screen printing techniques on PET or other flexible substrates. They can be also realized on clothes and on other kind of substrates, which are also biocompatible. The measurements are faster, they take 10 to 15 minutes. They require few microliters of sample, and the probe can be also changed depending on the analyte that we want to study. As a proof of concept, I present here the results obtained by exposing previously shown device to increasing concentration of ADAS spiked in a PDS pattern. Here we can see the change of the drain source output current is a function of the voltage of the gate. As we can see, we observe a change, a drift of the current, which is due to the different binding of ADAS on the surface. So by increasing the concentration, we induce a drift of the current. By normalizing the current differences of 8 different devices, we can obtain a signal which depends on the concentration of the analyte. We can observe a detection at really low concentration with a theoretical limit of detection of 100 femtomolar and a saturation of the signal around 500 picomolar. We can fit mathematical models on this data to obtain values like the constant of dissociation constant between the antibodies coupled, and we can also evaluate the different response against other anti-drug antibodies. For example, here there is a comparison between the exposure to the specific anti-drug antibodies and anti-drug antibodies against other drugs. As a perspective, we are developing a microfluidics system, so to have a constant flow of sample on the device. We are also fabricating these devices by printing technique on PET to reduce the cost of production. And also we are starting to test with real patient samples, which are more complex as composition. And also we are working on changing both the drug and light couple, also by studying the opposite detection, so by mobilizing the ADAS to study the presence of the drug in the samples. This work was performed in the Laboratory of Organic Electronics at the University of Modena, Reggio Emilia. Here you can see our website. The group is directed by Professor Fabio Viscarini, with the collaboration of Dr. Carlo Bortolotti and Dr. Marcello Berti. I want to thank also Professor Andrea Cossalizza and the other clinicians that work with the patients treated with immunotherapy, and also the funding that made possible this work by the project AMI, funded by the EURONanomed Free Scheme. I want to thank you for the attention, and please ask questions.
Thanks a lot, Matteo. Really nice. Can you hear me?
Thank you. Thank you very much.
Okay, great. I don't know if we already have some questions from the crowd. I have one. So imagine that we are like 5 to 10 years from here. What would be for you the most useful application? Would it be like for a portable, a wearable in patients that are treated with certain drugs for which you can have some resistance, etc.? Is it to do live monitoring? Is it to do screening for each patient for some personalized treatment? Do you see more like a tool for developing more efficient drugs or more efficient therapeutic protocols? What would be for you the ideal application of this technology?
So I think for this specific project, the idea is to develop point-of-care devices. So like we can go to the doctor, to our medic, or anyway, not in a specialized context like the hospital. And our doctor can put a drop of our blood in the device, and so we can detect biomarkers of a specific, for example, for biomarkers of cancer, or also detection of DNA. We can detect different analytes depending on the disease that we want to study. Also, in parallel, there is this other way to use these devices, which is the monitoring. Like there are already some devices that are able to monitor ECG, or just by putting the devices on the forehead, or also there are some devices which are able to do a continuous measurement of some parameters like heart rate. And the advantage is that these are really cost-effective devices that most of them are disposable. And it's interesting that we can produce them on some biocompatible substrates. Also, they can, in theory, be helpful in a preclinic development of some, for example, for immunotherapy, just to evaluate the potential development of anti-drug antibodies. So to have an evaluation of the possible side effects that can be generated also before the clinic, so in the development of drugs.
Okay, very good, and a very nice example of a cross key enabling technology development. I guess you need, and you will still need some collaborators, because you cannot mix all the technologies in a single place. I mean, you can, but maybe you will do some co-development. And that's a very, very interesting project. And well, I wish you good luck for the rest of the development of this technology. Thanks a lot.
Very much.
Okay, so now we'll move to speaker number 3, which is Vittoria Raffa from Università di Pisa in Italy. And she will speak about in vivo gene editing by nanotransducers. So Vittoria, if you can say hello while I'm loading your lecture.
Hello. Very glad today to share with you our results in the framework of iGene project, a project funded by the European Commission in the framework of Future Emerging Technologies Open Call. Well, iGene project would expand the therapeutic application of genome editing by developing new solutions to make therapeutic application of genome editing safer and more effective. Well, here the big challenge to address is the big dream of the gene therapy, which means the unique recognition of a short sequence among 3 billion base pair genome for editing and treating human disease such as cancers or genetic disease. Well, now we have the smart tool for doing this, the CRISPR-Cas9 system, the CRISPR-Cas9 molecular scissor, and of course a special thanks goes to the brilliant work of the scientist Charpentier at Douna that got the Nobel Prize in Chemistry a few days ago for this great discovery. Well, CRISPR-Cas9 is a big revolution in biotech, but now the open question is where we are for therapeutic application. Well, actually, we still have 2 open issues to solve. First, we know that at very low frequency, Cas9 could accept some mismatch in the target sequence, making possible off-target events, which means a wanted cut in the human genome, which of course raises ethical concerns. The second issue is related to the delivery of such a system within human cells. These concerns makes genome editing ethically acceptable just for few therapeutic applications, such as topical application in the human eyes for treating humanized disease, or ex vivo application for treating leukemia or other immunological disease. Okay, iGene project would expand the range of therapeutic application. Okay, the project proposes a light-inducible system, which is a nanoformulation specifically designed to solve the delivery issue, but also to make safer the editing process, thanks to an approach of programmable medicine. Well, if we talk in terms of Boolean algebra, the CRISPR-Cas9 system looks like a one-input logic gate where the output, the gene editing, comes true if the input is true, which means the locus recognition by the guide RNA. Well, the system we propose is a two-input logic gate where the output, gene editing, comes true if the two inputs, which are recognition, the locus by the guide RNA, and the like activation, makes simultaneously true. Indeed, by using a two inputs and logic gate rather than one input logic gate, the probability to get off-target events strongly decrease. Well, where we are, the project started last November, so during this first year we developed this nanoformulation, which is based on a plasmonic nanoparticle covalently linked to a modified enzyme. Then we tested this nano-Cas9 in vitro in serial experiments aimed to compare the nano-Cas9 with the gold standard Cas9 coming from IDT company. We tested the cleavage efficiency, and as you can see, we get quite similar performance. Indeed, we tested in vivo our nano-Cas9 by using for proof of concept study our model, which is zebrafish embryos. So we injected at one cell stage our nanoformulation. We have also post-control groups, and we target the tyrosinase gene. Why the tyrosinase gene? Because by performing the knockout of the tyrosinase gene, we have the loss of pigment in the developing larvae, which can be easily tracked in these embryos. Well, by comparing the nano-Cas9 with the control groups, again we got to the conclusion that our system can reach similar performance than the commercial Cas9 from IDT. Now we are in the stage to characterize the internalization mechanism of nano-Cas9 in human melanoma cell line, and it seems that our nano-Cas9 is able to spontaneously reach cell cytoplasm, but also cell nuclei. That's a very good indication for our scope. Well, in the next 3 years of project, we plan to perform light activation experiments in our model, which are zebrafish embryos and mouse model of human melanoma cancer, in order to understand the real therapeutic potential of iGene technology. Well, now time for to acknowledge people working in this project. We have the leukemia partners with expertise in material science, Istituto Italiano di Tecnologia with expertise in nanoplasmonic, the Lyonix company with expertise in lab-on-chip for biotech application, M-Square company which developed customized laser solution, and finally there is my team in Pisa with expertise in molecular biology and nanomedicine, and the team of Professor Pistello in Pisa with expertise in gene therapy. Before to conclude, I would dedicate a few seconds to introduce the University of Pisa, because University of Pisa is a beginner, joined this platform just a few months ago. Here in Pisa, we have a nanomedicine working group which is a network of scientists working in the University of Pisa in the field of nanomedicine, a network sponsored by the University of Pisa. I would acknowledge the scientific board, but also all the colleagues that belong to this group. We are more than 50 persons, 15 staff. So please visit our page for information. Of course, we are also looking for collaboration and networking activities. So if you are in the field, please contact us for collaboration. Thanks a lot for your attention. Special thanks to the organizers for this great opportunity.
Okay, thanks a lot and welcome back, Vittoria. Excellent talk. Before we move on, I would like just to say that it seems that we have some problems with the Q&A session during this meeting. Some people cannot ask questions. So no big deal. If you cannot ask questions in the Q&A, you can always raise your hand and we will give you the mic for your questions. And also another option is to write directly in the chat your comment or your questions. But now it seems to work back again, so that's good. We have a first question by Rosario Sanchez from Nanoquimbio, saying that you have done a very nice work and a very nice talk. What about scale up of this nano device? Can you give us some insight, please, Vittoria?
Yeah, we have in the consortium Prochimia, which is a company specialized in surface materials. So the task of this company is to scale up the production. So what we are doing is not to use commercial Cas9 enzymes. We are producing one mutated protein, so made protein. So we have all the, let's say, facilities to produce the protein, also to produce the nanoparticles. So everything is homemade. And Prochimia as a partner as the main task to standardize and scale up the production of such a system.
Great. Another question that I will double is from Alexandre Superville from ETPN, asking what would be the main application for this technology. And if you don't mind, I will add another layer on this question is, now that you have a new kind of logical gate to do some gene editing, we all know that DNA can be used in very, very complex manner now. Do you see some crazy applications like to do really programming, like genetic programming? What would be the fanciest application you can dream about by adding this new level of, let's say, computational engineering of DNA?
Yeah, these are two questions in one. So let's answer to the first one. So the first application, of course, is the easier one because we want to get results in a short time. So the first application is testing this technology on melanoma because this is a photoactivation. Of course, melanoma cancers, it's easy to be exposed to the radiation. So this is why we choose melanoma because it's a photoactivation. So you don't need the invasive approach for this photoactivation. So this was the first case study, but of course, there are a lot of applications that not necessarily cancers, also genetic disease, but let's try if this approach is feasible on surface cancer before to move to other applications. As for the second question, yes, of course, we have big gene. What we are doing so far, I discussed about 2 input logic gate. We're trying to do, and what we are testing is to implement multi-input, at least 3 input, 4 inputs logic gate to have, let's say, 3, 4 different conditions that comes through to have the output, the gene editing event. This is very challenging, and we are working on this concept.
Okay, great. I could imagine some applications, so I'm sure you have crazy things, but you cannot disclose everything. Victoria, that was really good. So we'll move on. We are right on time. That's good. We move on to the fourth speaker, which is Gil Gonçalves from University of Aveiro in Portugal. And Gil will speak about Graphene Quantum Dot Toolkit for Cancer Multiphoton Bioimaging. So you can open your mic and say hello to people while I'm loading your presentation.
Hi, good morning. My name is Gil. I'm working at the University of Aveiro, and I'm ready for sharing my presentation. Well, the title of my pitch is Graphene Quantum Dot Toolkit for Cancer Multiphoton Bioimaging. My vision is a world with more efficient and affordable cancer therapeutics that allow equal access to the treatment of all patients. The mission is design and implement innovative therapeutic solutions based on graphene materials for improved diagnostic and therapeutics of cancer patients. This project intends to transform a carbon material extremely cheap, graphite, with a cost of 0.05 euros per gram into a multifunctional nanomaterial with high optical performance, with much lower price than the current molecular markets that can reach up to 700,000 euros per gram. The last report of the World Health Organization showed an incidence of cancer cases of 80.1 million and the number of cancer-related deaths of 9.6 million, data from 2018. In 2030, it is expected to reach 30 million people deaths each year. The early diagnosis of cancer is the key to successful cancer treatment. However, conventional diagnostic methods still present significant technological limitations. X-ray, magnetic resonance imaging, computed tomography, endoscopy, and ultrasound are the current most cancer diagnostic techniques. However, these techniques only allow us to detect cancer when a visible change to the tissue occurs. At this stage, thousands of cancer cells may have proliferate and even metastasize. Moreover, current imaging methods are limited to distinguish benign lesions from malignant lesions. Cytopathology and histopathology are not effectively applied to detect cancer at an early stage. Nanotechnology is a powerful tool that can provide high sensitivity, specificity for the detection of extracellular cancer biomarkers and cancer cells, as well for in-vivo imaging. Our approach consisted to design novel graphene quantum dots with high 2-photon brightness for cancer bioimage, which allows suppressing the current 2-photon microscopy limitations for translation into the clinical practice, including the complexity of instrumentation and the lengthy data acquisition and analysis. The 2-photon excitation with nanometric resolution in the photo-induced process allows a deeper penetration and allows scattering in human tissue bioimaging. Graphene quantum dots have unique optical properties when compared with the most advanced luminescent biomarker in the market. Graphene quantum dots present a good water dispersibility and biocompatibility and a very versatile functionalization when compared with the water semiconductor nanodots. Graphene quantum dots usually present a luminescent dependent on excitation energy. The carbon particle surface passivation and doping with heteroatoms allow the carbon dots to obtain high fluorescence performance. In fact, carbon dots are already available on the market. The company Dots develops and manufactures carbon-based security markers and tracers with applications in lubricants, textile, polymer, agrochemicals, between others. Reported the large-scale preparation of graphene quantum dots based on the sole thermal treatment of graphene oxide, where the amide and amine ratio could be tuned by varying the concentration of hydrogen peroxide. The produced graphene quantum dots with an average size of 4 nanometers are characterized by the excitation-dependent emission, with the strongest emission peak at 510 nanometers at excitation at 206 nanometers. The excitation wavelength dependence of the emission, it is possible to distinguish two different regions. Region 1 for excitation above 350 nanometers, where the emission maximum shifts towards longer wavelengths, and the region 2 for excitation below 350 nanometers, where the position of the emission maximum is excitation independent. The quantum yield increased towards the long excitation wavelengths to a maximum of 11%. The highest 2-photon absorption cross-section estimated was 130 GM at 720 nanometers. There are two remarkable features in the nonlinear spectroscopy of these graphene quantum dots. First, The 2-photon excitation spectrum is a blue shift from the one photon excitation spectrum collect at same maxima. The graphene quantum dots show an early excitation independent emission peak at 550 nanometers up to photon excitation in the region of 700 to 1000 nanometers. The cell internalization studies show that graphene quantum dots have high propensity for internalization in cancer cells. The time-dependent cellular uptake, along with the exhibition of fluorescent signal with a vesicular appearance, suggests that this is mediated by activation of endocytic pathways. The next steps of this project will consist on optimize the size and atomic composition of graphene quantum dots, crystalline domains, and nitrogen doping in order to obtain high values of TPR cross-section, obtain fundamental knowledge of the structure, optical properties relationship, and translate this information to guidelines for the large-scale production of graphene quantum dots with nonlinear emission. Develop novel multifunctional graphene quantum dots that can combine diagnostics and therapy with high specificity to cancer cells by adding target agents. Our preliminary results that combine graphene quantum dots with theta-pyrolytic macrocycles show a synergistic effect on bioimaging and high efficient on photodynamic therapy. I would like to acknowledge to Dr. Paula Marques, Dr. Emilida Massoas, and Dr. Graça Neves, and all the other authors of this work, and to the Portuguese Foundation of Technology. Thank you very much for your attention. My contacts are below. See you.
Alexander, you are muted.
I know. That's the first technical problem. Hey,
yeah, it happens.
It's 4:46. It's okay for the first technical bug. Thanks a lot for the presentation. It was quite technical. It's not often very easy to follow for non-specialists. But I have 2 quick questions, and then maybe we'll have 1 question from the crowd. My first question is, well, you're working in a university and you plan to develop a technology that can have some indeed market applications for detection, for early detection of cancer. My question is very simple. Do you have any industry partner, very upstream, or even an SME, or do you want to create a spin-off if this technology works better? And second question is, have you already applied, and would you think about applying to the Health Tech Translation Advisory Board, the TAB, because we have experts that are fantastic to guide you in the development in such industrial applications?
Yeah, the first idea of this project is to reach the level of develop a spin-off for the application of these materials. However, in fact, there is some minimum requisites that you need to achieve in order to bring these materials for real applications. As I mentioned in my presentation, you need to increase the TPA cross-section, the optical properties of the system, in order to give enough resolution for your imaging. And another technical issue is that you need to increase the emission for near infrared region in order to be more useful in biological applications. But answer to your last question, I don't know that this is this type of institution that can support the project, but I think that could be a good idea to further develop these studies.
This is our job to make it well known. Yeah. The Health Tech Tab exists now for 6 years, and it exactly can help you with a vision from industry experts that can select and then guide you in the development of your invention to make it a potential successful business born and raised in Europe. And if you are interested in learning more, I really encourage you to stay until the very end of this event tomorrow, because in the conclusion of the workshop on nanomedicine translational issues, we will have one of the TAB experts, which is Mr. Eckhard Schwenner. He will be here to, let's say, share with you some lessons learned to help the best project to go faster to the market. And you can apply to the TAB anytime by just going on Google and have a look at health tech, like health technologies, health tech TAB, T-A-B. Thanks a lot for your great presentation, Jill, and all the best.
Very much.
All the best for the development of this technology.
Thank you. Bye.
Now we will finish with the last speaker. She comes from France. She's Ling Peng. She works for CNRS in Marseille, and she's a research director. And Ling will speak today about self-assembling dendrimer nanosystems for the delivery of drugs, nucleics, nucleic acid therapeutics, and imaging agents. So Ling, the floor is yours.
Okay, thank you. Hello to everybody. It's my great pleasure to present our work and share with you our results. I hope I can. Did you see my screen or I had a problem?
We see it, but it's the end of the diaporama.
Okay, I will try to check it again.
No problem.
Sorry.
We are exactly on time, so don't panic. Take your time and launch it correctly. You don't share your screen right now.
Yes, I will try to check.
By the way, you give me some space to recall that after that we will have the second keynote, but now it seems to work. Just go on presentation mode and feel free to start anytime.
Okay.
Thanks a lot.
So, okay, everybody can see. Okay, so I'm just talking about our dendrimer nano technology for biomedical applications developed in our group. I think most of you know that the nanotechnology-based drug delivery is very important in nanomedicine, and the principle for the size effect and the EPR effect everybody knows, so I will skip this. So one of our solutions is that try to establish self-assembling supramolecular dendrimers for drug delivery and for gene therapy and bioimaging. So this slide shows you the principle of our supramolecular dendrimer nanosystems. So the principle is very simple. We can use the amphiphenic dendrimers which self-assemble into the supramolecular dendrimers. And these supramolecular dendrimers, we can have the empty large void space in the interior, which can be used to encapsulate the hydrophobic drugs or image agent for drug delivery or for bioimaging. And also the surface, we can modify the surface to interact with nucleic acid for nucleic acid delivery, as well as for bioimaging, for carrying the imaging agent for bioimaging. So here is some of our proof of concept studies. So we try to develop these amphiphilic dendrimers, which self-assemble into the supramolecular dendrimers. As you can see, the size is around 15 to 20 nanometers, which is small for deep tumor penetration, but still large enough for having the EPR effect, as we can see here, using the near-infrared fluorescence imaging. We can see that the nanoparticle accumulate specifically at the tumor site. And here is the SPECT imaging, which we can see that the imaging probes also accumulate at the tumor place. And here is the PET image to show that all of these nanosystems can be significantly accumulate in the tumor via EPR effect. So they can be used as EPR biomarkers. And at the same time, also we try to elaborate the supramolecular dendrimer nanosystems for drug encapsulation for anti-cancer treatment. Oh, sorry. So you can see that if these nanomicelles can encapsulate the anti-cancer drug, here is doxorubicin or paclitaxel we have used. And there's also keep the small nano size around 15 to 20 nanometers, and they accumulate specifically at the tumor site, and they can significantly inhibit the tumor growth. Here you can see that this is a nano drug which encapsulated with the doxorubicin using our supramolecular dendrimers. And here is the control with BPS buffer and the free doxorubicin, which had no effect. This is a doxorubicin drug consistent cancer models. And here is the survival of the mice as you can see, all the mice which treated with doxorubicin almost died. Only one mice survived at the end, and all the mice survived when we're using our dendrimers to deliver the anti-cancer drug. And here is also the proof of concept studies using our dendrimer, amphiphenic dendrimers, which self-assemble into nanomicelles, and this nanomicelle will interact with this nanomicelle will be positively charged, and they can interact with negatively charged nucleic acid. In the here is the examples of the small interfering nucleic acid, and they form nanoparticles which will be enter into cells and then release the siRNA for gene silence. And we are very happy that our dendrimers works very well for gene silence in different immune cells. Here you can see that gene silence results in T cell, and here is the gene silence results in a natural killer cell, and here is in the monocyte and the macrophage. So we are trying to using these dendrimers, dendrimer delivery systems for siRNA delivery for gene therapy and immunotherapy. Finally, we can also using these supramolecular dendrimer nanosystems and to be decorated with targeting ligand. Here is the strategy which we used. For example, the siRNA dendrimer complex, which have the positively charged surface, theta potential, which can be interacted with the negatively charged peptide. This negatively charged peptide carrying the targeting group. So by decorating the nanoparticles with this peptide, we can have the nanoparticles with targeting peptide, with the targeting groups to target the cancer cells for targeted delivery, in addition to the passive delivery based on EPR effect. And here is the results that using the targeted delivery system, we can improve the anti-cancer activities much better than without the targeted delivery. So finally is that we are trying to working on using these self-assembly supramolecular dendrimer nanosystems for the different biomedical applications by combining the possibilities to carry both hydrophobic drugs and also hydrophilic nucleic acid, as well as the targeting possibilities for targeted deliveries. So we are working with different collaborators for this project. And we also we would like to establish new collaborations with people in this meeting and under the frame of European project. So that's all what I have. And thank you for your attention, and I will be happy to answer any of your questions. Thank you very much.
Thank you, Ling. Thank you so much, and for being very strict on timekeeping also. So thanks for that. That's very important for the quality of the event. So we have a question for someone called Ruth Schmidt. I don't know her. She said, you have now developed a platform. What are the next steps? Have you found a clinical indication that you want to focus on first, and where your platform seems to have the highest impact?
Okay, very good question. Actually, yes, we had active contact with the company which is developing siRNA therapeutics, and I don't know whether you know the Minatech, which has pushed the first small activation RNA in the clinic trial, and we have the patent about these dendrimers. Unfortunately, we are trying to for the license with and in collaboration with the companies try to synthesize our dendrimers for the GMP, but unfortunately this was stopped since the COVID situation, pandemic. So the company lost, will not right now invested on this issue at the moment, but we are still trying to contact different peoples and different companies using our dendrimers for further clinical applications. And also we are trying to set up a small startup with the oncologist in Marseille. This is just at the beginning, we didn't, that's the plan, but we haven't do that yet. Everything slowed down.
A little bit, just a little bit.
Okay.
Thank you for that. Just a last question from my part. I see, Spiros, that you have raised your hand. If you want to speak, I can open your mic and you can speak anytime indeed. But by the meantime, just a question. Do you see the main, in terms of technology, the main key advantage of your concept of this supramolecular assembly? Is it the fact that it is so easy more easy to produce? Or is it the fact that you can combine drugs and biologicals? What is for you the key interest of having these kind of supramolecular complex things when you compare it, like for instance, with a classical liposome?
Okay, first of all, for example, the classical liposome, usually they are toxic. For example, all the results we mentioned is with the pure dendrimer systems. We don't have any additives. So, for example, when we are collaborating with a biologist and always also ask why not you can use the lipofectamine, which works also. But for example, the lipofectamine, they cannot be used for in vivo. And you have to have the special formulation. And one of the advantage of our systems is that the formulation is very simple. We don't need to use organic solvent. And you just put it in the water and they can encapsulate the siRNA and also some of the drug, anti-cancer drug, hydrophobic drug. So it's quite, especially for the siRNA, for nucleic acid delivery, is very simple compared to what you heard about the lipid nanotechnology, which use the organic solvents. And our the delivery vectors is just put in the water.
Okay, without any gamer words, crystal clear. Thank you for that. Very inspiring, and we all wish you.
Can I ask a question?
Yes, please.
Ling Peng, thank you very much for a very nice talk. My question is related to why you chose to use these amphiphilic dendrons. I will not call them dendrimers, and not specifically synthesized amphiphilic dendrimers themselves. So you use, I mean, okay, in the title it says self-assembly, self-assembly dendrimer, but why don't you use something of the family of PAMAM or some of those? Okay. What is the advantage of this particular system?
Thank you very much. Very good question. Actually, we started with normal dendrimers, and the dendrimer synthesize. It looks perfect, but it's, in the practice work, it's very difficult to synthesize defect-free dendrimers. And also the reproducibility for higher generation dendrimer is very difficult. It's very challenging, technically challenging. And actually, a lot of dendrimers that have the activities for the delivery when they are in the high generation. So this is one of the problem to synthesize high generation dendrimers for biomedical application. Of course, if you want to publish the paper, it's easy. But if you really want to go to the biomedical applications, you have to have the problem with the reproducible synthesis and the characterizable synthesis. And actually, the first generation of dendrimers was scheduled for clinical trial with Minatec to deliver a small activation RNA. And because of the GMP production of the dendrimers in good quality, so the Minatec used the lipid vectors for the delivery of his siRNA delivery. And they told us that our dendrimers works really well. Although he cannot use our dendrimers for the clinic trial, but they use our dendrimers to publish the first results of the siRNA small activation RNA. So then we come to the question, so if we cannot make the high generation dendrimer, how can we with good quality, reproducible qualities, and in good in large quantity, then we come to the small amphiphenic dendrimers. And these small amphiphenic dendrimers is surprisingly well because they're self-assemble and they mimic the high generation dendrimers, not only in structures, but also in carrying the drug and the deliver drug and the siRNA delivery and the imaging agent. That's why we see they have particular interest and potential for further biomedical applications. And one of the other things I want to discuss with you is that they are not dendron. They are dendrimers because dendron means a part of the dendrimer. So dendron is like
yeah, maybe my word my choice of word was not absolutely correct. It's just I wanted to say that only one side is like a dendron or a dendrimer, and not
the dendron.
Not the other. So this way, yeah. Okay, I don't know how to call it. You know, you have a nice dendron in the photograph you showed us. Okay, thank you very much.
Thank you. Very good question. Thank you.
Thank you again. So it was not a bug. You did have a good question, Spiros. Thank you for that. And with this, we will close the first Pitch Me Up session. I will just stop sharing your screening. I would like to really thank very warmly all the speakers for being extremely, let's say, compliant with also the template that we have proposed. It was really easy and enjoyable to follow.